Hello, this is Mahanta Didia. This module on liquid dosage form of suspension is incorporated in the book on liquid formulations. In this module, we will look at quality and clinical study formulation. We started with physicochemical characterization of suspension, which are the scientific building blocks, then expanded it to pre-formulation, composition, and formulation, and manufacturing at lab and commercial scale. Throughout the development of suspension product, quality of the product must be consistent with the stage of development. Quality by design should be built in the development and product use in clinical study formulations through commercialization. The theme of this module is quality and formulations used in clinical testing. Please refer to module 2.3 for additional information on quality. And some of that information will not be repeated in this section. Quality has three components: quality by design, quality control, and stability. Suspensions are more complicated than monophasic solution. Insoluble particles form sediment from vehicle, which can lead to lack of uniformity of the active. Change in particle size and its physical chemical properties during manufacture or storage can lead to drug dissolution changes and impact on clinical effectiveness. Food and Drug Administration has a very specific guidance to examine quality of suspension products. Obviously, quality by design has been discussed previously. Just to refresh, it should be built throughout the development process, including production, testing, packaging, labeling, and distribution. QBD is a systematic approach. It has a predefined objective, product and process understanding. It also includes material attributes and is based on science and product quality risk mitigation. That means we have to identify potential risks and provide strategies to mitigate. QBD taken together with finished product quality control testing and stability ensures product quality in each lot. QBD critical quality attributes for suspension: physical, chemical, and microbiological. Physical: particle size distribution of the insoluble material, viscosity provides stable suspension, density, color, pH, zeta potential, flocculated suspension versus deflocculated suspension, sedimentation rate of insoluble particles. For pharmaceutical product, redispersibility of the product, uniform distribution of the drug, flavor, taste, palatability, grittiness can lead to CQA. In addition, dosing device, dose uniformity, whether it's a cup or a syringe, has to be demonstrated. And weight control at every major step during manufacturing. Packaged product, fill volume, weight control, both inverted and upright. Quality by design has chemical, microbiological, and biological activities. These attributes include active, assay, and identity, content uniformity to show that the proper dosing, impurities assay, preservative assay to show that product will be preserved through the shelf life. For pharmaceutical drugs, the solution of a drug, the packaging material needs to be compatible with the vehicle. In addition to that, microbiological attribute must conform to the USP or regulatory limits. Biotechnology products such as vaccine needs to meet the biological assays. Sterile product must be sterile, pyrogenicity needs to be measured, and osmolality needs to be measured. Let's look at product that was approved recently. Riluzole, 50 milligram per 10 ml. It's an oral suspension. It's a slightly brown, opaque, homogeneous suspension in 300 milliliter bottle and syringes used for dosing. This drug is used to treat the patient with ALS. It's also known in US as Lou Gehrig's disease. The drug has a sulfur nitrogen groups and fluoride groups. It's white to yellow powder and very soluble in methanol and very slightly soluble in water and sodium hydroxide. It has extremely metallic taste and is not palatable in a solution form. Suspension formulation, riluzole, insoluble solid, magnesium aluminum silicate, clay, suspending agent, sorbitol solution as a sweetener, bridge 20 as a wetting agent, purified water, saccharin sweetening agent, simethicone, sodium lauryl sulfate, and xanthan gum suspending agent. The instruction for Tuglutic bottle states that shake gently for at least 30 seconds before administration. 30 seconds is long shaking for a drug product. QBD risk assessment was conducted using guidances. Each CQA was identified, categorized as low, medium, or high risk at initial development, and strategies were developed to mitigate the medium and high risk so they all become low risk by developing critical material attributes, critical process, or additional specification for commercial product through the development. A study was conducted on palatability and consistency. Qualitative opinion for organoleptic characteristics of oral suspension OS was obtained. More than 50% indicated that original formulation in development was unacceptable from a taste perspective. So original development formulation had an issue. Consistency of the formulation was unacceptable in 23%. Less than one third considered consistency as acceptable. It shows that palatability was unacceptable. We concluded that these risks must be mitigated before product get approved. So QBD chart in the FDA file shows assay is acceptable, separation is acceptable, solid state, there are no polymorphic changes, dosing accuracy using the syringe was acceptable, palatability was not, leachable, extractable were not, the solution was acceptable, particle size of the active drug was not acceptable. Redispersibility was not acceptable, and content uniformity was not acceptable at initial development stages. Drug manufacturer provided the information to mitigate the risk, including 30-second shake well labeling. So redispersibility and content were addressed but the instruction to the patient. Stability data provided unopened bottle shelf life of 24 months. Upon shaking, 15 days of in-use stability was acceptable. Quality control is the second element in the quality of product. Quality control is a set of procedures in a lab for a specific batch to the established specific. Quality testing at each level of manufacturing needs to be done on representative sample. Specifications are based on CQA, clinical lot testing, pivotal stability lots, and validation of commercial products. Specifications are justified based on that. Example of a QC testing, USB monograph, and specification for acetaminophen oral suspension. Acetaminophen oral suspension, we looked at it in earlier module of commercial manufacturing process. USP test monograph. It starts with the limit of 90 to 110% of acetaminophen and that two reference standards, the drug and formulophenol, the degradant. Identification is done by filtration of a suspension, and filter is collected, and that's identified as acetaminophen. Uniformity of dosage for single unit container and deliverable volume for multiple container. Formulophenol degradant, and that needs to be tested. pH of the suspension ranges from 4 to 6.9. Assay methods are given here. It's an HPLC method. Let's look at the stability of omeprazole at 4 and 25 degrees. Omeprazole degrades rapidly at pH value below 7.4. That is, in acidic pHs, it's stable at alkaline pH of 9.5. So omeprazole is extemporaneously prepared suspension. Contains sweetener, glycerin, sodium CMC as a suspending agent, saccharin as a sweetening agent, bisulfite sodium as antioxidant, mint flavoring, sodium bicarbonate to adjust the pH to 9.5, and water purified is QS'd. Procedure: sodium CMC dispersion is made with glycerin. Active drug powder is hydrated with sodium CMC dispersion. Sorbitol is mixed with that to form preliminary suspension in separate container. The soluble material in water are added and sodium bicarbonate is added. One has to be very cautious with sodium bicarbonate not to have acidic environment. The final suspension containing 8.4% sodium bicarbonate is made and pH in the range of 9.4 to 10.1. We look at the stability at 4 and 25 degrees as omeprazole is unstable in suspension forms. The testings are appearance, palatability, pH, resuspendability, viscosity, assay, and microbial tests. Resuspendability included as part of stability test as a CQA. Viscosity is also CQA and included in stability testing. Palatability is done at initial stages at room temperature. Two different products, tablet and powder. In 30 days, assay only 10% of active is remaining. So it's stable at room temperature for 7 days. At 14 days, we start seeing the color change. Formulation B is not stable. The active degrades at 4 degrees. Formula A to meet the specification for pH. Resuspendability is then second. Assay continues to be acceptable. However, high assay of 109%, indicating the uniformity issue. At 4 degrees, formulation B, which was unstable at room temperature, shows non-stable 90 days and 4 degrees. This type of studies are conducted as pointed out in the earlier modules.